morning. Thank you very much for the very nice introduction and also for the invitation to talk here. My assignment here was an overview of the rice functional genomics project, the activities, and also the so-called Rice 2020 idea to share with you. And then for today's discussion, to get the community work together and also to gather the ideas and effort together, and so that we can work together as an international community. So many people have contributed to rice sequences here in the audience. And then after we get the rice sequences, the next question, actually at the time of doing sequencing, the question was, so what are we going to learn from rice sequences? So the functional genomics, the overall goal is to translate DNA sequences into information that are useful for biology, and also at the same time apply the information and knowledge and technologies in cultivar development. So based on what we learned from the annotation project, whatever release, so we have a huge amount of tasks in front of us. So genes and localizers are predicted for function or for protein coding, and plus many sequences we don't know yet for long coding. So the total amount of effort we need to make is huge. And then at the time when we started in China, with our functional genomics project, so we had constructed several components in our Chinese rice functional genomics program, which is a national special key project at that time. So we are intending to develop technical platforms and also resource platforms, identification and isolation of functional genes, and also functional genomics of agriculturally important trait. And at the same time, we do breeding applications. This is sort of the framework of rice functional genomics research in China. So we are constructing resource platforms and technological platforms like mutant libraries, at the same time germplasm collections, and then identify gene mapping, genetic analysis, and a lot of omics. So functional genomics under this proteomics, epigenomics, and transcriptomics, phenomics, and metabolomics, and also a lot of other things like bioinformatics, putting all this together. And eventually we'll get a regulatory network of the trait and also function of the genome. And applying all the information gained into our rice breeding programs. So the technological platform and the resource platforms we have developed, including mutant libraries, expressing profiles, and also genomics breeding, based breeding technologies, and bioinformatics. So the traits targeted for functional genomic studies include disease resistances, mostly blast and bacterial blight, and drought resistance, nitrogen and phosphorus use efficiency, plant growth and development, root system, grain quality, and yield and heterosis. So this is a construct we have been using for generating mutant libraries. So it has 3 built-in strategies for functional analysis of the rice genome, including insertional mutagenesis, enhancer trapping, and ecotopic expressions. All these 3 strategies seem to work very well. And actually, in the last 10 years, we have obtained a lot of results in every one of these strategies. So example of the mutant we collected based on student work in the field. So we could see mutations in the entire life cycle of the rice plant from the very early seedling stage, immediately after germination, all the way to maturity. And then we put all this together, make it globally available at rice mutant database, and at the same time distributing the collections and the seed stocks to researchers around the world. So this is a sort of a summary of the effort of the activities globally in terms of the numbers. So basically the tDNA insertions, TOR17, other transposable element, there's a number of lines with flunking sequence tags available. So the total number, if we put all this together, FSTs, and then is 2,4,600 plus sequences with uniquely mapped sequences, 211,000 sequences. So what is needed if we work as a community to saturate the rice genome with insertions based on the number, based on the annotations of the number of loci in the release. So something like 56,000 loci. This is something needed based on my own calculation is over half a million of the insertions needed to saturate the genome with the 99% probability. So we're less than halfway through yet. But the difficulty is huge already. So the number of insertion lines required for saturation is too large. That is very, very large. The workload too heavy for generating, maintaining, managing, and screening. And low proportion of good flunking sequences, and not easy to move the materials across the countries. So difficult to organize real meaningful international collaborations. So we need alternative approaches. Probably this is something we need to discuss after today's presentation. Another platform is expression profiling. So that is very useful and very important, and to trace the behavior of the genes and provide clues to understanding gene functions, and also help annotation of the genome, and also useful for construction of regulatory network. So one example we have done is using FOODCROPS gene chip to collect expression profile data for one hybrid, which is Xiangyu 63, which is the best hybrid in the hybrid rice history of China, and its parent, Minghui 63 and Zhengshan 97, from more than 40 tissues. So those are the tissues used for our expression profile. We obtain huge amount of data and perform data analysis and distribute the data into the database. Another platform that has been very useful is full-length cDNAs, collecting both one from the Japonica by the Japanese colleagues. Another is our effort in China, collaboration between Binghan and my own group. So from indica varieties, and totally this is what we did, and still continuing. We are working on Guanglu I4 and Minghui 63 and some other lines for full-length cDNA collections, which are useful for cloning genes and for proomic analysis. Another area is the cloning and characterization of important genes. So many of the contributors for the important genes published in the last few years are here, and we'll hear more in this meeting. So I'm only providing some numbers which we summarized last year. So as we can see how good we are doing in the community. So before the year of 2000, essentially we had very few genes. Since the starting of the functional genomic research, or since the obtaining availability of the right sequences, so the numbers are increasing very rapidly. So the total number, as of now, as of the time we made the review, was not very huge yet. It's slightly over 600. So then the year's effort of this year and later last year. So I would predict or guess the number is something like 600, 800, or probably not very much more. So according to the method we use for gene cloning, map-based cloning accounting for 27% of genes cloned, Gene tag mutant 21%. So this category is likely to grow in the next few years or in the next years. In the future, another big portion of the genes clone is based on reverse genetic approach. So overexpressing or antisense approach accounting for 52% of genes clone. So if we divide the genes clone according to the trait, we view that is as a breeder, we view the genes in a way according to its agricultural use, like yield, abiotic stress, disease resistance, nutrient use efficiency, grain quality, heading date, growth and development, root, and other traits. So this something related to yield account for the largest portion of the genes clone so far. And that is a genes clone. Another thing, as we have been doing, is so-called phenomics. So you probably all know there's a platform developed in Germany by Namlatech, but it's not very useful for rice work. So we in Wuhan, we have been developing a platform, a facility for phenotyping rice. So now the technology is developed by our own effort, and it's a collaboration between our university and the National Lab of Optics. So what happened was the facility just like some sort of a semi-automatic. So it could phenotype per facility 4,200 plants per day at the vegetative stage. And then what's more interest is it could produce scoring the yield trait, which is not available anywhere. I'm not sure, not aware of anywhere that has such facility to do that. We can get yield trait scores at 30 seconds per plant. It gives number of grains, field grains, and grain length, weight, and also yield per plant. And that is something already now in a stage of enlargement, expansion. Another effort we made in China is a resequencing of rice genome with new sequencing technology. So basically sequencing germplasm collections, which you are going to hear from Binghan and Kang today and tomorrow. And also another application of the technology is for genetic mapping populations. So it could create high-density bitmaps for QTL analysis. So one example is what Bingham published a couple of years back in Nature Genetics, sequence of 517 non-risk accessions from rice germplasm of China. So they could perform, actually could get a diversity analysis of rice germplasm and at the same time to do GWAS analysis of quantitative trait. So they could relate the DNA sequences to the performance, agronomic performance of the trait. Another effort we made is to develop constructing ultra-high density linkage map based on population sequencing, which is a population we have been working on for many years. But basically, there's a idea so-called maximum parsimony of recombination. So based on very little sequences of each line in the population recombinant imbrine line population, as little as 0.05%, 0.05 X coverage of the genome. And then that is basically the sort of steps in this genotyping. So very little sequencings, and then putting it together to identify the bins, and then putting together the genotype, the lines in the population, and it appears the accuracy of the map is very good. That can be confirmed, verified by the cloned genes. And then this map can be used for identifying QTLs for yield trait, and also any other trait you could phenotype it. And also, we use this map to analyze the genetic composition of heterosis in this particular population, which is elite hybrid, the hybrid parent. So we could get additive and dominance effect of each being in the entire genome. So we are trying to put things together in the at this stage, this phase of the project to develop information system. It's called Rice Information Gateway. We are intended this to be the rice version of TIRE, the Rapid Information Resources System. So in collecting the information and also analyzing information, providing the sources for the information for analysis, and helping the research community and putting the information together. Another effort is genomics-based breeding. I think now is the time. So we are making very big effort in whole genome selection for rice breeding. So that is my definition of whole genome selection is probably slightly different from what presented in elsewhere. So making use of genetics, functional and phenotypic information of the genes is based on high-throughput SNP detection technology, and a simultaneous selection for targets, non-targets, and genetic background at the whole genome level. And we hope to revolutionize efficiency and precision. So we are developing selection chip. So this is the first version, it's called Rice 6K. Now we have a second version, it's called Rice 60K. This is a testing breeding chip for rice. So another system we're also building is gene-specific. So gene-specific selection system for the targeted gene. In this case, we have been developing gene-specific system for blast resistance, for bacterial blight resistance, for brown plant hopper resistance, and also for grain quality. And one example is that we are doing selection for blast resistance breeding we are trying to introduce blast resistant genes, like in this case, a gene on chromosome 6 and 11. So we can, using this chip, we can see very clear how good the genetic background is and what is carried over from the donor parent. And then the lines in some area could be, we could use the lines and evaluate them in areas of production. So another notion, which is the theme for this meeting, is called the next green revolution. In China, we have a call for second green revolution. So second green revolution, by definition, 10 Chinese characters. So translated into English, less input, more production, better environment, which is very different from what is known as Green Revolution that occurred 40 years ago or 40 years back. So then in this context, we propose a notion of green super rice. Basically, we should apply less pesticide, less fertilizers, water saving, and drought resistance. So less pesticide means we need a lot of resistances to diseases and insects. Less fertilizers, we need a nutrient use efficiency. Water saving and drought resistance explains itself, and high yield and superior quality. So that has become something big. So we are working on that with very large teams. And this is one of the strategies for the development of green super rice. So targeting very many traits: insect resistance, disease resistance, nutrient use efficiency, drought resistance, quality and yield, using all kinds of technology. So using genes from non-rice sources, genes based on rice germplasm, and genes and technology based on functional genomics. Another strategy is to introduce a sort of introgressing of genetic diversity into the Chinese cultivar. So we started with 20 elite cultivars, grown very popular varieties in Chinese, in rice-producing areas of China, and using 500 plus accessions of germplasm worldwide, and we make huge amount of crosses and back crosses. And then at the very end, we would obtain very many near-isogenic introgress lines, and then evaluate them and for the trait of interest, and then try to identify the genes introgressed using new technologies, and then pyrimidine new germplasm to develop new cultivars. This has been successful, and many cultivars have been released from this project. So also a little bit touch on the concept of varietal designs. So in our view, there are 4 different levels, or 4 components, 4 elements in varietal designs. One is a yield limit to be achieved through a population structure that can make maximum use of the solar energy in a given ecological conditions. That is the utmost important. So the plant architecture to realize a population structure, that's a lower level, that's individual plant. So the third level is a trait to make up the plant architecture and also to achieve high quality resistances to multiple biotic and abiotic stresses and high nutrient use efficiency. And the fourth level is the genes to realize the trait and regulatory network to produce a trait, and also the breeding technologies for gene assembly. So 5 different elements we need to consider to put into the so-called varietal designs. And then touching on the theme of Rice 2020, that was my assignment to talk. It's a call for an international coordinated effort in rice functional genomics. And that was a paper we published in 2008 by the coordinators, coordinating committee of the China Rice Functional Genomics Project. So basically, we proposed international collaboration on 7 themes. So I'm not going to read them one by one, but basically we want to figure out the function of every gene. We want to sort of characterize, elucidate the regulatory network. We want to understand the natural variation of sativa and its wild relatives. And also we want to provide and put together information system, data management, and also for exchange and sharing, and also to facilitate the analysis of the community, and also establishment of a kit for high throughput knowledge-based rice breeding, so for breeding applications. And then we have another version now. I think Radwin is going to elaborate and going to talk and discuss about this. It's Rice 2020 Reverse, an urgent call to mobilize and coordinate rice functional genomics research worldwide. So we're going to hear more from him. So basically, that's what I have. I would acknowledge my colleagues in China. China Rice Functional Genomics Project is organized into 3 big teams. One team in Beijing, another team in Shanghai, another team in Wuhan. And also we have big teams in the Green Super Rice Project. The project right now is supported, it's becoming something big, supported by Ministry of Science and Technology in China, and also by the Gates Foundation for the international component, coordinated by Zhikang Li, who is here in the audience. So we are lucky to have support from a number of funding agencies. Thank you very much for your attention.
Kiva, you mentioned about the issues related to exchange of these mutants and tagged lines, and you said we have to think of an alternative strategy. Do you have some words about such an alternative strategy?
I'm not here from the stage.
No? You could not get it? I'm asking you, what is the alternative strategy you are thinking of for exchange of material, that is tagged lines and mutant lines?
I cannot hear you. Well, we had a Dr. Tayagi is asking how to share the mutant seed, so which has been very difficult. We have quite a bit of discussion, so we are willing to share, but it has been difficult. So we have a lot of quarantine requirement, certificate, permit, etc. So what we are doing now is whomever requested, we would provide seed. So we'll ship out the seed, and then without worrying about whether the individual is going to receive it. So we probably need to discuss about the strategy how to share the seed. That's very difficult anyway.